It is August 5th, Biology 105, Summer 25. This is the lecture. So we're here. We're in week 5. It's the last week. We finished our last lab yesterday. All right. We got a little bit of I'd say 3, maybe 4 successful PCR amplifications out of the entire class, which isn't bad. It's not that rare for us to get 0. Sometimes we get 100%. Remember, that lab involved doing a DNA extraction and setting up a PCR amplification. All right? So there's a lot of places where someone could go wrong. And you extracted straight out of, your cheek cells. So hopefully you got something out of it, seeing how these pieces can be amplified, understanding what gel electrophoresis is and what running a gel, making a gel looks like, some of the basic calculations that go into, doing that. And that's it, for labs. Today, we're going to look and see if any of the bacteria that we transformed, that should be living in that incubator right now, getting real stinky, we're going to see if any of them actually fluoresced. All right? Pretty cool stuff. And if you get a wild hair, and you'd like to, it's Tuesday, right? Yeah. You could, if we get any fluorescing colonies, take some, plate them, and draw a little picture. We'll let them grow, we'll let them incubate, and then they'll be a glowing little drawing that you can make out of transformed E. coli. If you want to do that, if we get successful transformations. After that today, all we're doing is lab final review. I'm going to go through all the topics that are going to be on the lab final. I'm going to open it up to questions from you, and once you're all done answering questions, we're all done going, and we're going to go home. All right? Or wherever you go after this class. In my mind, you all just sort of disappear into a mist, and then you reform in the mornings for my classes. All right? Cool. That's right. Okay, cool. I got it. A question, please.
We don't have lecture tomorrow.
Oh, lecture tomorrow. Yeah, there's lecture tomorrow. Yeah. Sorry. I don't want to get your hopes up too much. So we've got lecture on Wednesday, which is here. And then on Thursday will be the lecture final. So today's lab will be set aside for lab review. Then the lab will be the day, final will be the day after that. Wednesday, if we finish our PowerPoint ahead of time, I'll open up the rest of the lecture for us to do, discussions about study guide questions and things like that. I think I have like a 12, 15 appointments. I got a pretty hard out at our regular ending time. Anyway, any questions? I know we've talked a lot about the lab final, what the lecture final, and what to expect. But if you want me to reaffirm anything, I'm happy to do so. Study guides posted for the lecture final. Lab finals, open notes, open notebook. All right. Why don't we just go ahead and get back into our PowerPoint. Oh, and I'm going to be posting yesterday's lecture and lab today during our 5-minute break to get those up soon, since I know that we're kind of on a time crunch. I'll try to get today's lab video, lecture and lab uploaded today as well, but we'll see. Okay, what am I doing? Downloads. Week 5, where are you? Okay, we're talking about operons now, all right? So I'm trying to emphasize for you some differences between prokaryotes and eukaryotes here. If you remember, the biggest difference between the prokaryotes and the eukaryotes, right, is the presence of that nucleus. At least from our perspective, that's the most important thing, right? This allows both transcription and translation to occur simultaneously in the prokaryotes, while they have to occur in 2 steps in the eukaryotes. All right? This makes additional space for more regulation in the eukaryotic gene expression, where there's an additional stage of RNA processing, all stuff that we've talked about, okay? We're going to talk about one more aspect of prokaryotes and gene regulation. So We've talked about all this. I'm just going to kind of zip through it because we got to keep a healthy pace up. Transcription, translation, ribosomes, we talked about it all the different steps, polyribosomes. There's a video you can go back and watch. Okay, so now we're into regulation of gene expression. We're going to start with one aspect of prokaryotic gene expression regulation, and then we'll talk about eukaryotic gene expression regulation. Now remember, for a gene to be expressed, it needs to be transcribed into mRNA and then translated into a protein, okay? This promoter region in prokaryotes, in eukaryotes, no matter what you're talking about, the promoter region is very important because that's where RNA polymerase will bind. And RNA polymerase won't always bind unless there are transcription factors there to help it bind. There's a lot of regulation of gene expression that's just based entirely on will RNA polymerase bind to the promoter or not. Because if it won't bind, you can't get the mRNA. You can't get the mRNA, you can't get the protein. Cool? So now we're talking operons. An operon is a prokaryotic thing, okay, where they cluster related genes together so that they all can be transcribed at once, okay? So for example, we've got this metabolic pathway here where a precursor molecule gets converted into tryptophan. It takes 3 enzymes to do it. So you could see a case, or you could imagine a situation where you put those 3 enzyme genes together because you need all of them whenever you need to make tryptophan. So there's some signal that says, hey, we need tryptophan. Just make all the enzymes involved in the metabolic pathway. Bless you. All right? So an operon clusters related genes together. It still has a promoter region where RNA polymerase binds, but there's an additional piece of the promoter, okay? So this is within the promoter, and it's a region of the DNA called the operator. And the operator is going to further regulate this by either blocking RNA polymerase from moving forward and transcribing the genes, or by allowing it to move forward and transcribe the genes. Essentially, all right, in broad strokes, there's a thing that can bind the operator, and if it binds the operator, transcription can't happen. So now there's another step of regulation of expression here, where what regulates whether this thing is bound to the operator or not bound to the operator. Fair enough? All right, let's talk about it. 2 types of operons, inducible operons and repressible operons. An inducible operon is usually off, but can be activated due to a signal, okay? A repressible operon is usually on, but can be inactivated or repressed due to a signal. So let's think about tryptophan. It's an essential amino acid. You need it most of the time. Basically, you need it all the time. But if you've got a bunch of it, it would be great if you could stop transcribing those genes. So the tryptophan operon, and let's not get it twisted, the tryptophan operon thing is hypothetical. The prokaryotic genes for tryptophan production aren't necessarily in an operon. I'm using this as an example. Are we cool with that? It's just a hypothetical example. Okay. So like a tryptophan operon, right, would be inducible. I'm so sorry. It would be repressible, and it would turn off when there's enough tryptophan. It's like feedback inhibition, right? Where there's enough of the product, so let's just stop making the enzymes involved in producing the product. An inducible operon makes more sense if the enzymes are involved in breaking something down, like lactose or arabinose, okay? I believe the two example operons we're going to use are the tryptophan operon and the lactose operon. We'll see, though, in just a second. All right? And it's logical and it makes sense. Here's why. What's the consequence if the inducible operon can't be induced, can't be switched on? Well, you can't break down arabinose. You can't break down that sugar. There's still other sugars you can break down, other energy sources, so the organism won't die. But think about if you're not able to make tryptophan, if the repressible operon doesn't work, okay? Well, if the repressible operon, okay, can't be repressed, then you just have tryptophan constantly being produced. And that's a lot less bad for the cell than not being able to make tryptophan at all. So one way to remember what sort of operons are likely to be repressible and what sort of operons are likely to be inducible is to think about the consequences if the system doesn't work as intended. All right? And if the consequences are fatal, if the operon's repressible, then it's probably not repressible. You know what I mean? Cool. Let's keep on moving forward. I'll show you how this functions in more detail and hopefully have it make some more sense to you. And hopefully I'll stop stumbling over my words. You know, that would be nice, wouldn't it? Operon regulation. Okay, cool. So we've got a tryptophan operon. Good, good. We've got a lac operon. This produces the enzymes involved in lactose breakdown. So these are our two example operons that we're going to be using. Remember, tryptophan is an essential molecule. Lactase, or the lactases, those enzymes are breaking down lactose, aren't essential. They're nice if there's lactose present, because now you have access to another energy supply. But you don't need to be wasting time on producing these enzymes if there's no lactose present. Okay? Cool. Great. These are the molecules that are generated by the metabolic pathways, not pictures of the enzymes or anything. I mean, look, that's clearly lactose. You can tell just by looking at it that it's a sugar, right? Look at that glycosidic linkage, okay? Look at that C, H's, and O's, carbohydrates, right? I bet you it probably follows the C sub N, parentheses H2O sub N formula for its structure, okay? This right here is an amino acid. Look at the nitrogen in it. So, repressible operons, they are on unless turned off. Inducible operons, they are off unless turned on. Some generalizations, when you see tend to be, you know that there are operons that break these rules, right? Whenever something says usually or tends to be, it means that there are going to be exceptions. All right? But repressible operons, they tend to be pathways that build things up, or anabolic pathways. Because remember, that's an anabolic pathway. It builds something. All right? It uses energy to build something. Inducible operons tend to be catabolic pathways, pathways that break things down. Okay? And that makes sense because with the repressible operons, you want to be building the stuff until there's enough of it. And then you repress the operon and stop making the enzymes responsible for the pathway making the thing. With inducible operons, you don't want to make the enzymes unless their substrate is present. Does that make sense? Okay. Repressible operon, terminology, all right? Bless you. With repressible operons, the thing that binds the operator, okay, is called actually in both of them. Let me just be very careful with my language. I don't want to start on a bad, understanding here. In both inducible and repressible operons, okay, the thing that binds the operator is called a repressor. And when the repressor binds the operator, transcription can't occur. That's true of both. If the repressor is bound, okay, there's no expression of the genes. Same thing here. If the repressor is bound, there's no expression of the genes. The terminology that's different is in the molecule that regulates the activity of the repressor. It's kind of annoying, you know, but check this out. In a repressible operon, all right, the repressor is usually not bound to the operator. So usually, all the genes are being expressed. You're usually making tryptophan. There is a molecule called a co-repressor, which will bind to the repressor and help it repress. So the co-repressor binds to the repressor and allows it to bind to the operator. Okay? So these genes are making tryptophan, and that's great. And now you have enough tryptophan. Some of the tryptophan binds to the inactive repressor, acting as a co-repressor, and allows it to bind to the operator, preventing expression of those genes. So in a repressible operon, the molecule is called a co-repressor. In an inducible operon, the molecule is called an inducer, because usually, all right, the repressor is bound to the operator, and therefore there's no gene expression. But now, some lactose is available. And you're like, oh, cool, there's lactose here now. We better start transcribing those genes so we can break this lactose down. So some of the lactose can act as an inducer, bind to the repressor, and cause it to fall off of the operator, allowing expression of those genes. And once all the lactose is broken down in the system, there's no more inducer present, and the repressor can bind again to the operator, shutting down production of those genes. All right? This makes sense so far? Yes, please.
The inducer is in place of the co-repressor?
Yes. So it's really frustrating, right? Because both a co-repressor and an inducer bind to the repressor.
Okay.
It just depends on, with an inducer, when it binds to the repressor, it causes the repressor to fall off of the operator. But a co-repressor, when it binds to the repressor, it causes it to attach to the operator.
Okay, same but opposite.
Yeah, that's exactly it. Same but opposite.
Okay.
Yep. Yes.
Are operons only in prokaryotes?
Yes.
Okay.
Yep. There's the, because of the RNA processing step in the eukaryotes. Yeah. Yeah. Cool. So check it out, right? Here's the lactose operon in action. Allolactose is just a form of lactose. Don't worry about it. It's present when other forms of lactose are present. Okay? It's an inducer. So when lactose is present, one form, allolactose, binds to the repressor protein, okay, inactivates the repressor. All right? Cool. There's another gene responsible for making the repressor protein. That's something that you don't have to really worry. You should think about it, right? The repressor is a protein. So sometimes there could be a mutation in an organism where the gene transcribing the repressor protein gets mutated and doesn't work. And now the operon can't be regulated anymore. Does that make sense? If you lose the repressor entirely. So, or you could have a mutation that causes the repressor to be unable to bind to its co-repressor or inducer. You see what I'm saying? Like if this site didn't work, now the repressor's always bound to the dang operator, and the genes can't be expressed even when the inducer is present. Just something to think about, all right? So the genome is regulating itself, or it's making the proteins responsible for its own regulation. It's all crazy stuff. Here's the system. Let's look at it. Repressor, normally it'd be bound, but right now there's lactose present. Lactose binds to the repressor, inactivating it. Therefore, it's acting as an inducer. When this repressor is inactivated, RNA polymerase can zip along and produce this long mRNA strand, which now has all of the genes, all the instructions for protein synthesis in sequence. Okay? So there are start and stop codons. Ribosomes bind here, start and stop, and start and stop. And you can make 3 of the enzymes, or these 3 enzymes that are necessary for using lactose. So the operon, remember, is the cluster of related genes and the promoter region, which also includes an operator region. Okay? Cool. And yeah, in fall or spring, I'd have like 5 more slides about this, but I have to cut something somewhere. Yes?
So, since you said transcription and translation occur simultaneously, this is technically preventing transcription.
This is technically preventing transcription, the production of the mRNA molecule. But if there's no mRNA molecule,
No translation.
exactly, there's nothing to translate. Points. Very good. Okay, so this is the only aspect of prokaryotic gene regulation we worry about. All right, is operons. Of course, there's more gene regulation stuff going on in prokaryotes, but we're just giving you one piece here. How do we feel about operons? Are we comfortable moving on to eukaryotic gene regulation? Okay. Eukaryotic gene regulation is more complicated. Everything in the eukaryotes is more complicated. So there are all of these steps, all right, in gene expression in the eukaryotes, okay? And all of these steps are opportunities to regulate and make changes. So check this out, right? First of all, a eukaryotic genome, everything's packaged around these, everything's wrapped around these histone proteins. You saw them previously in the video. We've talked about them a little bit. This can control which genes can actually be read. So remember the example, like this is now a linear strand of DNA, right? And it's got instructions written on it for how to make a protein. But if I take part of this and wrap it tightly around a histone protein, well, now I can't read the message. The machinery can't attach if it's wrapped too tightly around a histone, so transcription translation doesn't happen. But you can make that thing get looser or move the histone downstream or upstream, and now those instructions can be read, okay? So this is an example of epigenetics, all right, where there are changes to phenotype, changes to gene expression patterns that didn't have to come from any changes in nucleotide sequences, all right? The nucleotide sequence is still the same, it's just some of it was inaccessible. So you can regulate access to the gene itself. Histone acetylation and methylation, all right, are the 2 processes. In general, one usually makes things more expressed, one thing makes things less expressed, but we're not going to worry about a lot of these details. I want you to focus more on the idea that, oh, if we make it impossible to access the gene by wrapping that portion of the chromosome around a histone protein, then it can't be expressed. Cool? All right. Here you go. The DNA is wrapped around histones. And yeah, usually if you acetylate the histones, right, if you add these groups, it'll make the protein loosen its grip on the chromosomes, on the DNA. It's not something that you need to worry a ton about. Okay? But epigenetics is incredibly important. It's just very complicated, so we kind of breeze past it and make sure that you're aware that it exists. All right? So epigenetics are recognized more and more and more as incredibly, incredibly important as a source of changes in phenotypes. All right? And epigenetic changes can be inherited. There's evidence to show that. So you can inherit the same sequence of DNA, but you could also inherit different epigenetic patterns. It doesn't matter if you have the DNA if you can't read it. Just things to think about. Just float them out there. Okay? Next, let's assume the gene is actually available for transcription. Well, transcription regulation. Okay? Transcription factors, activators, repressors, all of these things exist in DNA as well, or in eukaryotes as well, okay? So we've got transcription factors, all these things about distal control elements, don't worry about. Inducers, repressor, or activators, repressors, they do what it sounds like they do. They activate or repress expression. This is genetics stuff for you to worry about in your genetics course. I want you to be thinking about, oh, in the eukaryote, there are all of these points where expression can be regulated, okay? So in general, right, these transcription factors need to be present for RNA polymerase to bind, but there are other proteins involved. There's this, usually the DNA does this weird thing where you have this distal control element where activators get bound to an enhancer region. Don't memorize these terms. They're going to take up space in your brain you need for other stuff, okay? And then the whole thing bends with this DNA bending protein, and then these mediator proteins facilitate the attachment of RNA polymerase, and it forms this thing called the transcription initiation complex, and it's all super complicated. But it allows for more tight regulation. If there's one thing I want you to know here, it's transcription factors, okay? You need the transcription factors there to help RNA polymerase bind. That's in-depth enough for our Bio 105 understanding, okay? Cool. Next, RNA processing. We've already talked about it, all right? Alternative splicing, how you can how you can splice these things and you can shuffle exons around, right? All that can be cell type specific through regulation of which spliceosomes are present, those special enzymes that chop up the immature RNA, the primary RNA transcript, and produce the mature messenger RNA, okay? Things that I've already mentioned verbally to you when we talked about RNA processing. But here, you've got a figure that's trying to bring it all together about all these different steps. Okay? So now the RNA has been processed, and it gets exported into the cytoplasm. Now, there are ways to regulate gene expression even at this point. You can degrade that RNA. Okay? So, somebody asked a question about this either yesterday, it must have been yesterday, because this is only Tuesday. Like, what controls the amount of time that the RNA stays stable in the cytoplasm? Somebody asked. Does anybody remember asking that? Whoever it was, it's a great question, okay? That's also regulated. So, in the instructions for making the RNA, there can also be instructions for essentially like, this is going to be more stable RNA, so it can be translated more. Or this is going to be less stable RNA, so it's going to be translated less. All right? Often controlled in the untranslated region. All right? So it's kind of like a telomere on an RNA molecule. Kind of. All right? Helps to stabilize it, determine its half-life. Okay? But remember how RNA is a single-stranded molecule? Well, microRNAs, RNA interference, siRNAs, all of these things are also designed to stop the expression of RNA. I'm going to keep it very basic. All right? Here's the RNA strand. It's a single-stranded molecule, okay? Now, another gene produces a small piece of RNA that's complementary to the single-stranded RNA and binds right there. Well, a ribosome doesn't know what to do with that. A ribosome now can't do anything with this piece of RNA. So you effectively have shut down the production of that protein. It's all complementary, so it's just going to bind there. MicroRNAs, RNA interference, SI, small interfering RNAs, SNPs work kind of like this. All right. Sometimes these work just by binding to the single-stranded RNA and preventing the ribosome from being able to read. Sometimes these work by binding to the single-stranded RNA and serving as a target for proteins that'll chop up the RNA and degrade it. All right? The CRISPR-Cas9 system works like that. So that works for silencing genes by destroying the RNA. Kind of interesting stuff. Cool. It's specific, targets them for degradation. Here's microRNA happening. All right, microRNA binds to target mRNA. It can either be degraded or just block translation. Because it needed to be single-stranded to work. Cool. All right. Oh gosh, I'm actually very embarrassed. I just told you, the CRISPR-Cas9 system works like this, by interfering with mRNA translation into a protein. And as I'm saying that, I'm like, that's inaccurate, John. That's wrong. You've lied to the students. CRISPR-Cas9 isn't as hot as it used to be, you know. It was like the big hot topic for a while. They made like 6 episodes of a television show that was all about CRISPR-Cas9 gene editing. So I don't know if people still are as excited about it, but that's not how that works. So we can talk about it later, maybe during break or something, because we got so much to get through. But do you kind of understand why microRNAs would interfere with gene expression? It needs to be single-stranded for the ribosome to work. So you make a piece of DNA that's complementary to the mRNA, but just to a small piece of it, and then the ribosome can't read it because it's designed to read a single-stranded structure, and now it's double-stranded. All right? How do we feel? It's all right? Okay. That's all we talk about with gene regulation, okay? Now we're into mutations. We're going to talk about these mutations are small changes to the nucleotide sequence. All right? Usually it involves you added in an extra nucleotide by mistake during DNA replication, or you missed a nucleotide during DNA replication. All right? Or you substituted a nucleotide during DNA replication. And it changes the sequence of nucleotides. These are mutations, okay? So we're going to talk about base pair insertions, deletions, substitutions, silent, missense, nonsense mutations in general. So originally, right, all of these mutations were the source of variability within the population. Something goes wrong during S phase of interphase, and you accidentally make one strand different than the other. So sometimes what can happen is you make the complementary strand, it's different and no longer complementary to the parental strand, and the DNA repair machinery fixes it the wrong way and swaps out the template strand, you know? So we're going to talk about small-scale mutations. We're going to focus on those. Mutations are changes to the nucleotide sequence, all right? There can be larger scale mutations that can happen, but these are the most common that can just occur because the DNA replication machinery is not perfect. It's just a bunch of proteins floating around in goo, you know? All right. Two broad categories. Point mutations. What's a point mutation? A point mutation is when a single nucleotide is affected. It's either added in, or it's removed, or it's replaced with something different. Make sense? Okay, so we have single nucleotide pair substitutions when there was an A and a T here, and now it's a C and a G, all right, after replication. Single nucleotide pair substitutions. Then we've got insertions or deletions where a new nucleotide gets added into the sequence. I've repeated this, I know, many times already. I just really want to make sure we're all comfortable with this so far. Because now we're going to look at the reading frame and how different types of mutations affect the reading frame. Reading frame's really important. Okay, I've already talked to you about it once, but it's easy enough to forget that. Okay, so check it out, right? Remember, this is what we've got here is a sequence of RNA. You can tell it's RNA because there's a U in there. Makes it easy, right? It's being read in this direction. There's no directionality on here, but it should be a 5' end here and a 3' end here, you know, if you want to be accurate to the way that the codons would be read. Normally, in this example right here, the reading frame goes like this. AUG is start, then CCC encodes proline, then AAG encodes lysine, and so on and so forth. But imagine that this nucleotide isn't there, okay? Right? And you start reading here. Now you've got UGC as the first codon, which includes cysteine, let's just say, it's cis something. All right, then CCA, then AGA. You'll see here it's an entirely, or not entirely, but it's a very different sequence of amino acids. The third reading frame, if we start reading here, codon, 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 also a different sequence of amino acids. So anything that shifts the reading frame can be very harmful for the protein that gets produced. It's very harmful for the gene because it changes every single codon downstream of where the change occurred. Question?
So that means that insertion and deletion is more drastic and bad than the substitution?
100% correct. Points for you. Absolutely. 4 of them. All right. I didn't think I gave numbers for the points earlier. The other person, you also can have 4 points, whoever that was. I gave points to. I'm sorry for not quantifying them. Very good. That's why insertions and deletions are usually more harmful than single nucleotide pair substitutions. Because a substitution here, right, for something else, sure, it might make this amino acid different, but all the amino acids downstream are going to be fine. Okay? Now, sometimes a single nucleotide pair substitution could do something really bad, like turn a codon into a stop codon, and now the protein was supposed to be this long, but it's actually just this long, and it probably doesn't work, because now there's a stop codon there, you know? So they can all be harmful. All right. Nucleotide pair substitutions. One nucleotide and its partner get swapped with another one. All right? C and G becomes A and T. Or the T and the A kind of switch sides, you know, that can have the same effect. Silent mutations are mutations that don't change the protein structure at all. Okay? A silent mutation doesn't change the protein structure due to the redundancy in the genetic code. Remember how multiple codons, different codons can encode the same amino acid. So like CCC, CCG, CCA, CCU, they can all encode the same thing, possibly. All right? Therefore, any of those switched out nucleotide pairs, any of those changed sequences are silent and have no impact on the organism and its phenotype. All right, make sense? Those are the silent mutations, the ones that do not impact amino acid sequence. Missense mutations, okay, these are not frame shift mutations here because they're all the result of substitutions. So missense mutations, they change the amino acid, but the protein's 240 amino acids long. One amino acid changing might have very little impact. All right? So we call it a missense mutation. And it's usually not as harmful as the third one here, and that's the nonsense mutation, which is what I mentioned just a moment ago. In a nonsense mutation, you end up with a stop codon where there used to be a regular amino acid codon. And now the protein's getting made as usual, and the ribosome goes, oh, stop, fall off, I'm done. And it made, what, a fifth, a tenth, 90% of the protein? Who knows? But usually that makes a non-functional protein. Sometimes it can make a protein that has an undesired function, even. All right? These mutations can be bad. Here's a big figure thing. I really like to make sure that we understand all the text stuff because this can be overwhelming once it just pops up on the screen. All right? But trust me, it just summarizes everything. It just summarizes everything that was already written down. And everything that we've covered so far, logically, does it sort of make sense to people? You understand the codons, the frame shift mutations, the point mutations, the base pair substitutions, why they're not as harmful. It's all good? Okay. I feel like we kind of are pretty comfortable with all that. Let's look at this big figure here. All right? Here's the wild type DNA. So this is the normal DNA, right? Here's the sequence up at the top. Over here on this left column, we've got different nucleotide pair substitutions. And over here on the right column, we have different nucleotide pair insertions or deletions, and some of the potential outcomes of these. Okay? So look here at this substitution. It happened in the third nucleotide of the codon, changing GGC to GGU. Both encode for glycine, so it did not affect the amino acid sequence. It did not affect the protein sequence, and therefore the mutation is silent and it has no real impact. There can be impacts on things like, say, the stability of the DNA molecule due to the number of triple or double bonds, hydrogen bonds, but all of that is unimportant to us, okay? Next, we've got the missense mutation here. In this case, we got a TA instead of a C here, which corresponds to right there, all right? So that's a nucleotide pair substitution, okay? T instead of C, A instead of G, right there. Leading to, in the RNA, all right, a different codon. Instead of being GGC, it's AGC. And now we end up with serine instead of glycine. So this has changed one amino acid. It is a missense mutation. The protein may still function normally, or it may function not quite as well, all right? But it's probably not going to be as drastic as, say, a nonsense mutation. This is an extreme example where the second codon right after the start codon, there was a mutation in the DNA encoding it, right? That turned AAG, which encodes lysine, into UAG, which is a stop codon. So now the ribosome attaches to this mRNA and goes, start, stop. Not going to be a functional protein, okay? Nonsense mutation. Cool? Now let's skip over to the second column, all right? What you've got here are 2 frame shift mutations, one resulting from insertion of an extra nucleotide, one resulting from deletion of a nucleotide. Now, this frame shift mutation here, I hate that they do this in this textbook figure. This frame shift mutation here ended up generating a stop codon, making it also be a nonsense mutation, where you got a stop codon where there would have normally been an amino acid codon, right? Here's a frame shift with a deletion, and look, methionine, lysine, and now you got leucine and alanine instead of these two, because you got an insertion here, it changed the reading frame, and all the codons got shifted by a single base. All right, solid enough. Here, we've got a 3 nucleotide pair deletion, which can happen. And if that happens, that's usually way less harmful than a single nucleotide insertion or deletion, because it just makes you lose 1 amino acid instead of changing every single amino acid downstream of the mutation. How are we feeling? Okay. That's our mutation talk. Not all mutations always have a phenotype, okay? So remember our enzyme, oh, actually we didn't do, wait, no, yeah, we did use polyphenol oxidase. We did the potato lab. Remember our polyphenol oxidase enzyme is responsible for generating pigments, all right? In cats, they've got this enzyme called tyrosinase, okay? And what it's responsible for is attaching pigment to fur, basically. All right? Now, some cats have a mutation in the gene that changed the amino acid sequence, but it still left a functioning protein. The protein's just less stable. All right? So you can have, and so it's probably a frame shift mutation, you know. You can have a mutation that doesn't affect the entire protein, right? Just some of the amino acids. So the protein still works, the enzyme still works, but the enzyme is less stable, okay? So in this case, the tyrosinase enzyme in cats with this coloration, all right, is less heat stable. And so at the core of the cat's body, closer to the main mass of the cat, it's a weird way to talk about an animal, closer to the cat's tummy-tum, all right, it's warmer and the enzyme doesn't work. So the cat is white there. At the extremities of the cat, where the temperature is colder, okay, the enzyme functions, all right? So that's how you get this sort of coloration. So it's not active at higher body temps, but it's active in paws, tails, ears, the little nose, all that stuff. All right? Cool. So this is an example of, well, it's an example of missense mutations, but probably a frame shift mutation near the end of an mRNA molecule to make the enzyme less stable like this. Cool? Sick. So it's a mutation that has a phenotype in some parts of the organism, but not all parts of the organism. So is it a silent mutation? Not really, but in the tail it is. That's all of our mutation stuff. Okay. Now we're into the real, right in the last week, with a day and a half of lecture left, we're into the genetic stuff that you've probably seen before and that you absolutely have to know, okay, for future genetics courses. Core terminology, all right, how patterns of inheritance work, but none of it makes sense unless we talk about the cell cycle and DNA replication and how mutations can occur and all this other stuff. This is going to feel like a pretty drastic shift right now into new material, but it's tied to all the other material because it's also all about the genetic material. It's like a joke, kind of. Okay, now, I'm going to try to get everybody's brains back into thinking about chromosomes and less about nucleotides. All right? And we're going to refresh on a little bit of terminology here, things like homologous chromosomes. You got to think about meiosis and how it works a little bit here. Okay, otherwise all these patterns of inheritance don't make sense. Solid? Okay, so just remember, here we are. Let's look at the terminology. Ask me questions if they pop up. Homologous chromosomes, okay? Alleles, different versions of the same gene. All right, the alleles should be at the same location on the chromosome. Following meiosis, you split up those homologous pairs, okay? You reduce ploidy, you generate haploid gametes. One gamete fuses with another gamete in fertilization, generating a diploid zygote, which then divides via mitosis to make the organism. Now, in this new organism, you've got one paternal and one maternal form of the gene. They may encode the same protein, all right? They might. They might both be the same form of the gene, even though there can be maybe small differences in the nucleotide sequence. Those all could be silent mutations, okay? Or missense mutations that don't actually affect the functioning of the protein. The sequences are likely to be slightly different between the two chromosomes now, all right? But they might make fully functioning proteins. Therefore, they encode the same phenotypes. Does this make sense so far? Stupendous. I gotta stop saying it that way. That doesn't really prompt you as students to actually ask questions if I keep saying, everybody understands this, right? That's bad. It's bad teaching. What I should say is, are there any questions about this? Can I provide any clarification here? It's much more encouraging, I think. So I'm going to switch over in the last week to doing that more. Are there any questions about this? Yes, please.
So,
It worked. I got you. I'm deducting points. Please.
so what you're referring to in terms of like possible changes in sequence, that's because of stuff that happened in the original homologous chromosome.
In this organism, yeah.
Okay.
And also, you could have already had them. You inherited them from your parents, right? So you inherit these alleles, these gene versions from your parents, and maybe you have a non-functioning form of a gene. And then you pass that on to your offspring. And now they have that non-functioning form of that gene. Sometimes that's good. Sometimes that's actually adaptive, to not make a certain protein. Sometimes it's bad. All right? It's maladaptive. Good, bad, those aren't statements that we need to really stick to. Okay? But yeah, the key thing here is that they're slightly different forms of the same gene. It's actually pretty unlikely that a gene on one of your homologous chromosomes and a gene on the other homologous chromosome have exactly the same nucleotide sequence. There's probably a couple of little changes in there. But are they drastic enough to actually make the protein that's produced be any different? And so for that to make sense, that's why we kind of seg from mutations and silent mutations into this topic. Yeah, probably a little more drawn out than you needed, but solid? Yeah. Awesome. Any other questions on this so far? Okay, very good. We're going to talk Mendel's P experiments, all right? One brave man who challenged himself to see how much urine he could consume in a single day. Mendel's P experiments ended in disaster when his glass jars shattered in the earthquake of 1748. I don't know why you all hate fun so much, but I decided to make that joke, all right? We're going to talk about Mendel's experiments, the rules that he derived, the theories that he derived, and Mendelian genetics, which is basically just like simple patterns of inheritance, all right? The fundamental processes that make Mendel's laws work are the law of segregation and the law of independent assortment, okay? Which dictate that you only inherit one copy of each allele from each parent, okay? You just get one copy of the gene from each parent because homologous chromosomes separate, all right, during generation of the gametes. And these are the same allele, and they're going to separate because these are copies on the sister chromatids. Listen, it makes more sense if you remember all the meiosis stuff, but it can make sense if you don't, you know? All right. And the law of independent assortment, which says that each chromosome, right, is inherited independently of every other chromosome. So genes that are on separate chromosomes are inherited independently. So which allele you inherit from the parent on which allele located here does not determine which allele located here you inherit. All right? So in general, Mendel says like, oh, all right, you get one copy of the gene from one parent and one copy of the gene from the other parent. Why does that happen? Because of the process that occurs at meiosis 1. He generates the law of segregation for that, all right? That the two genes that were present in each parent segregate from one another, and you receive only one of those two alleles that were present in the parent. The law of independent assortment, he developed, saying each characteristic is inherited separately of each other characteristic. He didn't have the language or the ability to look at chromosomes or anything like this. So he's like, yeah, the characteristics are inherited separately from one another. But we now know that's because of independent assortment during meiosis 1, during metaphase 1. All right? Question, please.
So just to make sure I get it right, for humans as the example, each law applies to like one of the 23 pairs that we have?
Each law applies to the whole set. Because,
like,
yeah.
The whole set, but like the law of segregations, like we always get for pair one from mom, one from dad, and then independent assortment is like the two chromosomes for pair one, they sort independently.
Yeah. And then you just apply that exact same statement to every other chromosome. Right?
Yeah.
Yep, you got it. So chromosome 1 gets split in mom, split in dad, right? Chromosome 1 could be like this, or like this. Chromosome 2 could be like this, or like this. Chromosome 3 could be like this, or like this, in mom and in dad, right? In the microgamete generating individual and the megagamete generating individual. Since that way we can apply it to, all diploid organisms. Yes, perfect. Cool. Yeah, no problem. How do we feel about this slide? It's going to come back, and we're going to talk about it in more detail. It is, however, time for the 5-minute break. So I'm going to pause here, and then we're going to get into Mendelian genetics. All right? Okay. Lecture continues August 5th, 2025. We are at slide 79. We need to get to slide 150 by the end of the day tomorrow. So, it's possible. Let's talk Mendel. All right? Let's talk Mendel. Let's talk where John put his clicker. That'd be a great thing to know. Did I set it in the bathroom? Did I put it right here? I did. Here it is. All right. Very good. Gotcha. Ha ha ha. Blasted your eye. All right. Mendel, monk, failed teacher, could never do what I do. All right. Now, he wanted to be a researcher and an educator. Went to a school for that. Didn't work, so he went into God stuff, God work. Yeah. A lot of people did that. Science, religion, very tightly intrinsically linked, all right? people like Mendel wanted to understand how it all worked. What were the mechanisms for, getting the variety of life that we see around us. He made it a bunch of peas, all right? And he tracked what they looked like. He saw that, hey, there's a bunch of different varieties of pea plants. They have distinct characters. You can control their mating easily enough by just snipping off the different parts, the pollen-producing or the egg-producing parts, all right? They reproduce quickly, so he could do a lot of work in his life, all right? And each flower has both sperm and egg-producing organs. All right, pretty handy. What did Mendel do? Well, he took some, removed the sperm-producing parts, all right, so that they would not self-fertilize, and then used other plants, took pollen from them, and directly applied them to the stigma where the pollen grains could germinate, and then the pollen tube could grow down in and fertilize the eggs within the carpels. All right, this is not a botany course, so you don't need to know the parts of a flower, okay? You don't need to know the anthers or the stamens or the carpels or the stigmas or the andrecium or any of that stuff. All right? So, check it out. He takes a purple flower, takes a white flower, goes, okay, cool, I'm going to breed these and see what happens. All right? So here's Mendel's experimental setup with some important terminology that you must know. All right. The pea generation. All right? It's not because they're peas. It stands for parental generation. The pea generation is made of true breeding individuals. What does that mean? It means that, well, in practice, you keep breeding these purple flowers, and you're like, well, these always make more purple flowers, all right? But from a genetic standpoint, it means that these individuals only have the purple flower allele. And these individuals only have the white flower allele, making them true breeding, all right? They will always pass on that allele, okay? Does that, are there any questions about that? Caught myself. Yes?
So always homozygous?
Oh, gosh, I'm just a tsunami of points for you, okay? Absolutely. They're homozygous, yep. So this individual is homozygous for the purple allele, and this individual here is homozygous for the white allele, all right? 2 copies of the purple, 2 copies of the white. Very good. So when you breed 2 true breeding parents, you'll make an offspring that's always heterozygous, because it gets 1 copy of the purple allele from this parent and 1 copy of the white allele from this parent. Mendel found that, all these plants had purple flowers. Now we know that that means the purple allele is dominant and the white allele is recessive. The purple allele, you only need 1 copy of it to express the purple phenotype. Then Mendel took this generation here, this generation being called the F1 generation. F stands for filial, if you care. These are hybrids, okay? These are heterozygous individuals right here, the F1 generation, and breeds 2 of the F1 generation, 2 individuals, or just self-pollinates and takes the pollen from within a single, flower and fertilizes itself. You know what I mean? You generate the F2 generation, and the F2 generation has this 3-to-1 ratio of phenotypes, okay? 3 purple flowers to every 1 white flower. So that indicates a lot of stuff, right? If you don't know that genes exist or that chromosomes exist, right, it indicates that, the unit that passed on a white flower characteristic was still here in these purple flowers. All right? So you can see how Mendel started to develop those laws. He's like, this must have inherited 1 unit of purple and 1 unit of white. And then when I cross this, okay, with another one, well, some individuals inherit 2 units of purple, some individuals inherit 1 unit of purple and 1 unit of white, and some individuals inherit 2 units of white. Therefore, we get 2 possible phenotypes. All right? Okay. Now, why does this happen? All right. It all has to do with meiosis. So if we've got this individual right here, uppercase P, lowercase p, this individual is heterozygous, okay, and expresses the dominant phenotype being purple. It can generate 2 possible gametes, a lowercase p and an uppercase p. These are the 2 possible gametes that it can generate following meiosis because of the law of segregation, right? You only inherit 1 allele from each parent. So each parent only donates 1 of the alleles that they have. How do we feel so far? Any questions about this? Now, think about this. The likelihood of inheriting these alleles, 50-50 in the offspring, right? So you've got a 0.5 probability of inheriting this allele and a 0.5 probability of inheriting this allele. Then you have the other parent. You have a 0.5, 0.5 probability of inheriting either allele. All right? There are 4 potential offspring that you could make here. A homozygous dominant, a heterozygous individual here, a heterozygous individual here, and a homozygous recessive individual here. Now, note that you can get a heterozygote in 2 different ways. All right? You could either inherit the recessive allele from this parent and the dominant allele from this parent, or the dominant allele from this parent and the recessive allele from this parent. Okay? Also, this is all just probabilities. Okay? So what this is showing you is there's a 50% chance of being a heterozygote if you breed 2 heterozygotes. There's a 25% chance of being homozygous dominant here, and a 25% chance of being homozygous recessive. And check this out. 0.5 times 0.5 equals 0.25. All right? The probability of each one of these events is 0.25. Okay? This is a Punnett square. You've probably seen them. You've probably had to use them. It's just a way to visualize the probability of independent events happening together. Okay? Because this, which one of these alleles you inherit, is independent from which one of these alleles you inherit. Each one was random. All right? How do you feel? How do we feel? Any questions about this so far? Punnett squares are a trap. All right? They are so easy and simple when you're doing basic genetics, and you can use them all the way through BIOL 105. And then you're doing a tetrahybrid cross in Biology 340, where you have to track 4 different genes, all right? And they all have different conformations. Nothing's heterozygous. And now you're trying to make, what, like a 128-cell Punnett square? You're not going to do it. So I'm going to try to help you avoid that trap by, from the very beginning, explaining this in terms of probabilities. Because it's all just probabilities. And Mendel wrote about it like that. The mathematics of probabilities already were understood and pretty clearly expressed. Okay? So why can't we understand it at that same level too? We're going to try. All right. This here is what we call a monohybrid cross. You're crossing two individuals that are heterozygous for a single character that you're tracking. Here are the phenotypic and genotypic ratios. Remember, the phenotype is the physical expression of the genotype. So the phenotype here is either purple flowers or white flowers. The genotypes are either homozygous dominant, homozygous recessive, or heterozygous. Both of these genotypes lead to the same dominant phenotype, both the homozygous dominant and the heterozygous. Only the homozygous recessive genotype leads to the recessive phenotype. Are there any questions about genotypes and phenotypes so far? Usually, you use this convention where the alleles will stand for the phenotype, P for purple, and the dominant allele will determine the letter. So capital P for purple, the dominant allele, and then lowercase p for not purple. All right? Kind of makes sense? Cool, cool. So if we were tracking alleles for red flower color, we might do capital R for the red, lowercase r for the not red version. So let's look again, little zoomed in with more details. P generation, purple and white, they're true breeding, so they're both homozygous. Very good job. The gametes, well, this individual can only make a dominant. This individual can only make a recessive. So the probability of receiving this allele is 1. You will receive this allele. And the probability of receiving this allele is 1. The probability of both events happening together is 1 times 1, which equals 1. 100% of these offspring should be heterozygous. Just probability math, all right? Nobody ever does this as a Punnett square. You could. Well, why would you? All right? You never have to use Punnett squares starting today with the introduction of them. All right, here we go. This individual here, half of the gametes it makes will be dominant, have the dominant allele. Half of the gametes it makes will have the recessive allele. All right? This individual's the hybrid. It's the heterozygote. If you breed one heterozygote with another, well, the probability of receiving a dominant allele from each is 1/2 times 1/2, or 1/4. The probability of receiving a recessive allele from each is 1/2 times 1/2, or 1/4. The probability of receiving a dominant allele from one and a recessive allele from the other is 1/2 times 1/2 plus 1/2 times 1/2 equaling 1/2. All right? Sick. So there it is as a Punnett square, but it's just probabilities. Okay? Cool. Awesome. Here are the big characteristics that Mendel tracked. Flower color, flower position, seed color, seed shape, pod color, stem length. The first year I taught this course, I explained every single one of these phenotypes. It was not necessary. I think that all of these make sense, right? These are the phenotypes he tracked. And he found that, yeah, each one of these characteristics followed those patterns of inheritance, all right, with one phenotype being dominant to the other, and doing two true breeding parents together to get a monohybrid or a hybrid, and then making a monohybrid cross. You found the same 3-to-1 ratios of phenotypes, all right? So these numbers right here are the ratios of the number of dominant phenotypes to the number of recessive phenotypes, right? So check it out, 705 purples to 224 white flowers, all right? there should be, based on probabilities, roughly 3 purples per every 1 white flower. There should be, based on the probabilities, roughly 3 axial flower position plants to every 1 terminal flower position plant. All right? But it's just probabilities, so it's not going to be exactly 3 to 1, but it's going to be close, you know? Now, let's say that you were trying to determine whether one of these characteristics followed the rules of a dominant versus recessive allele. All right? I wonder if there's a statistical test we could use to determine that. I don't have to wonder, because there is. Let's take a look at this example right here. This is a bit of an aside, but I'm going to do it to try to tie in all the big stuff we've been talking about. Check this out. All right? Calculator. This is my favorite app. It's super fun. It's got all these games that you can do on it. So check this out. Flower position. 651 plus 207. Okay, 858 total individuals. Well, if you did a monohybrid cross, you would expect that 3/4 of the offspring should have the dominant phenotype. 643.5. So our expected number of individuals with the dominant phenotype is 643.5. Our observed number of individuals with the dominant phenotype is 651. There are 2 possible outcomes, meaning degrees of freedom equals 1. What am I describing here? Chi-square analysis, right? So you could totally do that to see if the observed value was different from the expected value and see if the inheritance patterns are following the rules of dominant recessive genes. Anyway, that was an aside, but I think an important one because it just, ties everything back together. Whatever. We got 2 days left, Are we going to learn anything new really today or tomorrow? The answer is yes. We're going to learn so many cool things. Check this out. How could this actually work? Well, if we're talking about classic dominant and recessive alleles, it's often this simple. One allele encodes a functioning enzyme, and one allele does not. There's some mutation that makes the protein not work. All right? So check this out. This enzyme is involved in producing purple pigment. The purple allele makes the enzyme. The recessive allele does not make purple pigment, the white flower allele, okay? Therefore, the flower appears white. Nice. That's why we use color usually as the example for this Mendelian inheritance stuff, because it's like the most simple. You get a pigment or you don't get a pigment. And in the heterozygotes, you have one copy of the gene that makes the pigment, or the enzyme that produces the pigment. So you still have enough enzyme to make the pigment. Are there any questions about this? That's good. Okay. Mendel's model. There are these alternative versions of genes called alleles. He didn't call them genes. He didn't call them chromosomes. He called them either factors or particles. He used language like that. But this is Mendel's rules that he developed with modern terminology sort of pasted onto it. All right? So alternative versions of genes account for variations in inherited characters. These are the alleles. For each character, you get 2 alleles, one from each parent. All right? You know that this isn't going to always be the case with organisms that aren't diploid, but we don't talk about that. If 2 alleles differ, one determines the phenotype and the other has no noticeable effect. This only works if you've got a truly dominant and a truly recessive gene. All right? That's common, but it's not always the case. Sometimes there are a bunch of different alleles for a gene, and many of them encode something that functions slightly differently. So it's not going to behave like this. It's not going to follow the same rules. But right here, when we're talking about like classic Mendelian genetics, all right, we are talking about truly dominant and truly recessive alleles, all right? Hey, Ashley, everybody say, hey, Ashley. Truly dominant versus truly recessive alleles, okay? Where, awesome, one copy of the dominant allele is enough for us to express the dominant phenotype, all right? And the recessive version of the allele doesn't really do anything. All right, so these are Mendel's rules, and there are things that break Mendel's rules. But Mendel's rules laid the groundwork to begin understanding genetics and to make better predictions of patterns of inheritance. Okay? So the last one here is each type of allele is inherited separately from each other type of allele. All right? The genes for flower color are inherited separately from the genes for pod shape or flower position, all right, or pod color, okay? Cool? Awesome. These are the new rules. One, don't pick up the phone. You know that song? Man. That feels like a current reference to me. That's really sad. It's like Dua Lipa. I got some rules to tell you. 1, alternative versions of genes account for variations in inherited characters. 2, I don't know, what did I do to make you all hate me so much? I don't understand it. We used to have so much fun in this class. We're just tired. It's okay. All right. Look, here's, the king in this alien civilization beginning to understand the rules of random segregation, all right, crossing over events, all allele combinations. He's starting to get it. All right? Well, they're starting to get it, really, since they're all asexually reproducing organisms on this planet in the stuff of life. Check it out. Catch the fever of the stuff of life. Now, this rule, all right, that these things will be inherited differently from one another gets broken by more complicated patterns of inheritance, which is going to be a topic that we talk about, all right? So don't worry. Of course it gets more complicated. Here are 2 homologous chromosomes with 3 loci being indicated. In this diploid organism, the diploid organism is homozygous dominant for gene P, homozygous recessive for gene A, and heterozygous for gene B. Are there any questions about this, what I'm trying to illustrate for you here? Please. Oh, no, you're just saying hi? Hello. Yeah. Are there any questions here about what I'm trying to illustrate with this? There's not one gene per chromosome, right? And you can be homozygous dominant for some genes, heterozygous for others, homozygous recessive for others. It may go without saying, or maybe it's good to clarify. Let's just keep our pace up. We're hitting a pretty good stride right now. If we keep this up for the next half hour, whoo, we're gonna have some time for studying tomorrow. All right. Monohybrids, monohybrid crosses, okay? Mendel derived the law of segregation, okay? That being that you only receive one version of the gene from each parent through monohybrid crosses, okay? He generated individuals that he knew were hybrids for a given gene, all right? He generated heterozygous individuals, and then he crossed them in what's called a monohybrid cross. You're tracking one characteristic, and the parents there, or the F1 generation you're crossing, right? The individuals you're crossing are hybrids for that one characteristic. Monohybrid cross. So in a dihybrid cross, you would be crossing in organisms that are heterozygous for 2 characteristics. All right? And then tracking what their offspring look like. Okay. So by tracking the results of monohybrid crosses, Mendel writes all these numbers down, says, oh, there's 3-to-1 ratios. Well, that would work if the individuals were inheriting one version from each parent, and one version was good enough to express the dominant phenotype. All right? So Mendel just had this to work with. It's pretty incredible. He had this. And this is the data. This is it. All right? And he derived these rules by understanding mathematics and probabilities. All right? Cool? Okay. Law of segregation, boom. Law of independent assortment. If you're going to determine that the different genes are inherited separately from one another, right? You got to track multiple characters. So it just makes sense that he derived the law of independent assortment, okay, that one allele is inherited independently of the other ones. Is my language for that working for people? Can I clarify that in any way? He did that, but yes, please.
So character, is that just like another word for trait?
Yes, yes. The character, the phenotype, the trait. Yeah. So, for example, flower color is inherited independently of flower position. All right? Now, to develop that rule, you would have to track both of those. So what he did is he generated dihybrids, individuals that were heterozygous both for flower color and flower position, and then tracked both of those characters in the offspring. All right? And just like the offspring, you got to keep them separated when you're talking about the law of independent assortment. That's a reference as well. Just going to go heavy references today to things. You know, I saw the offspring in concert once. Pretty cool dude right here. Yeah, they pulled a piano out on stage. Remember Ex-Nay on the Ombre, the album where they got all sad? Then there was also the song about the kid who picks up what he thinks is a candy bar and it's feces. Remember that? That cassette tape made me cry when I was a kid because it's got a song about how he misses his dead significant other. And so I would sit in the back of the car and weep over this cassette. There you go. There's some information about me and about the offspring. You want to keep talking about the law of independent assortment now? Thank you. Me too. All right. Check it out. So, if the genes are on separate chromosomes, yeah, they get inherited independently. All right? Sometimes there's crossing over, and that can complicate things, where it makes it look like the two characters got inherited together because a crossing over event happened. All right? If they're on the same chromosome, they can be inherited together. If they're very close to one another on the same chromosome, it's likely they'll be inherited together because if a crossing over event occurs, you're going to get both of the genes. Okay? So anyway, law of independent assortment works when the genes are on separate chromosomes. Okay? Because each chromosome gets independently assorted, independently aligned during metaphase 1 of meiosis. Cool? All right, here's a dihybrid cross. So when I was a student, I didn't want to learn the probability math. I said, I'm not going to do it. You can't make me do it. I didn't say that out loud to the teacher because I was shy, you know? But I just said, whatever. I'll just always draw a Punnett square. Sure, you can do it with a dihybrid cross, but it's a mess when you're trying to do an exam. Horrible. You'll have to do a dihybrid cross question basically in your final, okay? And you can do it with a Punnett square, but it's going to make your life way more complicated, Just use the probability math that I'm going to teach you today. Okay? Here's a dihybrid cross. Check it out. This parental generation, the true breeding generation, they are true breeding for round peas and yellow peas right here. This individual is true breeding for wrinkled peas and green peas. All right? The Y allele encodes fruit color. Well, seed color, right? Cool. You cross these individuals, you must receive dominant versions of each allele from this parent and recessive versions of each allele from this parent. You generate the dihybrid. Now, Mendel made a prediction that if these were inherited together, you would expect that 3-to-1 phenotypic ratio, with 3 of the dominant phenotypes to 1 of the recessive phenotypes. All right? But this is not what happened. Instead, he found a 9-to-3 to 3-to-1 phenotypic ratio, where 9 of the individuals were dominant for both phenotypes. 3 of them were dominant for one but recessive for the other. 3 of them were recessive for one but dominant for the other. And 1 out of every 16 individuals was recessive for both phenotypes. All right? Remember, these are probabilities and ratios. It doesn't necessarily mean that he bred exactly 16 of them and 9 of them were round and yellow. He bred thousands of them, produced hundreds of them, you know. Yes, please.
So the being round and yellow, so these two phenotypes are dependent on one another? Does it work like?
They're inherited independently of one another. They're not dependent on one another is the key thing here. Yeah. Because if they were being inherited together, like as a little package, all right, then you would expect to have 3 individuals dominant for both characters and only 1 individual recessive for both characters, because they're linked, so it should follow these rules. But it didn't follow these rules when he did these dihybrid crosses. That led him to say, oh, they're being inherited separately. Because he basically set up all these different probabilities, all right, like what's the probability of each one of these gametes, and then found, oh, the laws of probability, if these are inherited independently of one another, predict really close values to what I observed. Close ratios to what I observed. Yeah, pretty cool stuff. Awesome. So, there are 9-to-3 to 3-to-1 is the golden phenotypic ratio for a dihybrid cross. All right, that's what people usually will memorize there. All right, for the genotypic ratio, which was 1-to-2-to-1 in a monohybrid cross, we don't memorize it for a dihybrid cross because it's an annoying number. All right, but 9-to-3 to 3-to-1 is the phenotypic ratio. Now, check this out, okay? If this clicks for you, it's great. If it doesn't, it'll click eventually and you'll wish that you'd listened to me. All right? Each one of these cells represents a genotype that arose in a different way due to a combination of possible alleles. The sperm cell has a 1 out of 4 probability of having a dominant and a dominant allele, okay? So the probability of also receiving a dominant allele from the other parent in the egg cell is 1 out of 4 times 1 out of 4, or 1 out of 16. So 1 out of 16 offspring will inherit dominant alleles from both parents. That's all the Punnett square is meant to allow, is to make it, so you can visualize this. It's a visualization tool of probabilities. But you can just mathematically figure this out. It's not the most difficult thing in the world to determine all the different possible genotypes that come from a single parent, right? Let's say we've got Oh, we're doing R's. I could have just R, Y. The 4 potential gametes. There's that potential, that potential, that potential, and that potential. All right? You've got a 50% chance of inheriting either one of these and a 50% chance of inheriting either one of these. Wait a second. 0.5 times 0.5 is 0.25, which is 1/4. So actually, this right here is also just the probability of each one of these independent events happening. All right? What do I mean? How do you get dominant here? Well, there's a 50% chance of receiving the dominant R. There's a 50% chance of receiving the dominant Y when you generate a sperm. Meaning 0.25. 1 out of 4. Okay? 50% chance of getting the recessive, 50% chance of getting the dominant, 0.25. Just probabilities. Okay? We'll do some practice problems where I really show you this in practice, and at least for my learning style, that works the best for me. Hopefully it'll work for you as well. But of course, I'm happy to answer any questions that you've got based on this Punnett square of a dihybrid cross. Yes, please.
There was phenotype and would genotype be the other one where it's a certain number, like in the middle, would that be, it's not 16, is it?
So in this case, you'll note that there are some of these outcomes lead to the same genotype, right? So there's not 16 potential genotypes, right? There are 16 potential like allele combinations, but sometimes it's really frustrating because it's all probabilities, all right? The genotypic ratio, I'm just going to ask you not to worry about it in the dihybrid crosses, all right? But the genotype for this individual is homozygous dominant for round, homozygous dominant for yellow. The phenotype is round and yellow. This individual here is homozygous recessive for round and homozygous dominant for yellow. The phenotype is wrinkled yellow. Does that make sense? Solid. We cool with all that? Sick. Good deal. And you can predict the likelihood of producing any genotype or any phenotype, all right, by understanding the probability of the events that lead to that genotype or that phenotype. All right? Yes, please.
What's the nomenclature when you're describing heterozygous and homozygous for when it's like RR, lowercase y, lowercase y?
Yeah. Oh, yeah. When you're talking about like multiple characteristics.
Should I not worry about it, or?
No. It's totally fine. You just talk about each character separately.
Okay.
You say it's homozygous for this, heterozygous for this. It's homozygous dominant for round, heterozygous for yellow. And then, if you're tracking like 5 characters, you describe the genotype for each one. Then you can do the phenotype more quickly usually. Yeah. Yes.
I think I'm putting together the answer. Is there a way to do this with, was it Harvey Weinberg, where you're adding multiple traits together? That's part of the chi-square thing where there's like how many factors are you including in your calculation?
So you could predict, right? Let's say that you did this experiment and you made 1,000 P's, right? You would predict that 9/16 of them are this phenotype, 3/16 are this phenotype, 3/16 are this phenotype, and 1/16 are this phenotype, right? Those would be your expected number of individuals. Then you would compare that to your observed number of individuals with degrees of freedom equal to 3 because there are 4 potential outcomes. And that's how you could say, ooh, these characters, if they are being inherited separately and following the law of independent assortment, well, then these are our expected values. If our observed values differed from that, you would say, oh, well then, in that case, these characters aren't being inherited independently. Maybe they're linked to one another. Maybe they're on the same chromosome, for example. All right, so you could use chi-square analysis for this absolutely, all right? Because remember, there are other types of inheritance that don't follow these rules. Yeah, solid.
Can you imagine what the equations would look like? So if we have the p squared, 2pq, and then q squared, how would you get the fourth term? Would it be like two rounds of the heterozygotes?
No, you don't even have to do that. Yeah, because that p squared, 2pq, q squared, that works when you're tracking a single allele, right? If we're trying to say, hey, now if you're trying to track, are these alleles evolving from generation to generation, that's a very different question that doesn't require anything on this screen. There, you would probably just track each one independently. You know, you would have an observed, an expected, you get your allele frequencies figured out, then you do your, genotypic frequencies that you observed versus what you expected. But you can use a chi-square in this case to not determine if these alleles are evolving, but to determine if the alleles are being inherited independently. So a chi-square analysis can be used anytime that you have an expected value and you want to see if your observed values are different than your expected values, which I think is a better learning outcome, is a pretty big learning outcome actually, because you'll be surprised how often you find yourself in that situation, where I know what I'm expecting, my observations are a little different than what I expected, but is the difference statistically significant? It's a really powerful test, the chi-square test. Yeah. Awesome. Okay, so here's the slide, the image from the Stuff of Life that talks about this, the dihybrid cross, okay? Genotype assortments, there's all the offspring, it's cute, they're like all leaving, okay? They're all walking off of the Punnett Square, okay? So, it's all right here in the Stuff of Life as well. I'm not gonna read these, okay, standing in front of you. It's not a very good use of time, which you're, paying for. But the slide is there if you don't want to download the PDF, and if you don't want to read the book, if you don't want to buy the darn book, some of the key images are up there. My intention is just to show you that, yes, the Stuff of Life goes over this as well. Anyway, so Mendel called them particles. Those particles are genes, all right? So we just call them genes, we call them alleles. Like, there's different versions of particles. It's like, okay, fine, all right? All of this is governed by the laws of probability. Mendel used math to figure out all these rules. All he had were observed numbers of phenotypes. That's all he had, all right? And based on a basic understanding of probabilities, he was able to figure out that, okay, there's some particle that's being inherited here. Some particles coming from the sperm, some particles coming from the egg. And oh, look, in this case, there's two separate particles, and they're being inherited separately. All right? Now, what stage of the cell cycle of meiosis leads to these two Mendelian laws? Well, the law of segregation happens because of anaphase 1, when homologous chromosomes are separated. That means gametes are only going to have one form of each particle, of each gene. Even if anaphase 2 didn't work, it's the same allele on either sister chromatid. Okay? Is that a hand? I thought it was a hand. No? Okay, cool. The law of independent assortment, that's because of independent assortment of each homologous pair of chromosomes at metaphase 1, okay? There's a 3-to-1 ratio in the F2 generation of monohybrid crosses. That allowed Mendel to determine that the law of segregation was happening. And the dihybrid cross of unlinked genes, 9:3 to 3:1 ratio, allowed him to determine that the alleles are being inherited independently of one another. All right. It's all probability. Let's talk probabilities. No, where's my probabilities? Where's my probability? No, they're not until here. That would have been a way smoother transition. All right. It's all about probabilities. Tomorrow, we will delve deep into probabilities. But today, let's sort of finish by introducing some more important information about how we use the predictions that Mendel generated, and also about the types of inheritance that break Mendel's rules, leading to unexpected results. All right? Cool. So one way to utilize Mendel's predictions, all right, is to do a test cross. It's done in very unethical animal breeding all the time, where they'll be like, well, we don't know if that individual is homozygous dominant or heterozygous because it has a dominant phenotype. So what you do is you breed it with an individual you know is homozygous recessive, and then you see what the offspring look like. And if they have any offspring that are, well, homozygous recessive, then you know that the parent had a heterozygous genotype. So check this out. Here's an individual with a dominant phenotype. It's got purple flowers, but you don't know if it's homozygous dominant or heterozygous. Here's an individual with a recessive phenotype. You know its genotype is homozygous recessive. If the purple flowered parent is homozygous dominant, all the offspring will be heterozygous. There is a 1 probability of receiving a dominant allele here, and a 1 probability of receiving a recessive allele here. Therefore, all of the offspring will be heterozygous and have the dominant phenotype. In this case, if the purple flowered parent is heterozygous, there is a 0.5 probability of receiving the dominant allele and a 0.5 probability of receiving the recessive allele, but there is a 1 probability of receiving the recessive allele. That means 0.5 probability of heterozygous, 0.5 probability of homozygous recessive. All right? It's all probability math. The Punnett square just visualizes probabilities. Cool? Any questions about that? So a test cross can let us determine the genotype if we don't know if an individual is homozygous dominant or heterozygous. And Mendel would have to do test crosses to determine that he really had true breeding parents, right? For that P generation in those crosses. I've never sat down during a lecture in here, but my goodness, this is exhausting. Pedigrees, all right? Where do you fit in pedigrees? Here, I guess, all right? I had to put them in somewhere. I put them in right here. A pedigree is a family tree. It describes, usually it indicates phenotypes. Sometimes it indicates genotypes. All right? It allows us to trace inheritance patterns. So right here, okay, the shorthand is square indicates male, circle indicates female, a filled-in symbol indicates presence of the trait of interest. All right? So this is a pedigree chart that tracks widow's peak, okay? A filled-in symbol means there's a widow's peak present, all right? Now this pedigree chart has been annotated with genotypes, but usually a pedigree chart will just have phenotypes. So here's a male with a widow's peak, here's a female without a widow's peak, here are their offspring. Here's a male with no widow's peak. Here's a female with a widow's peak. Here are their offspring. Here are all their genotypes. Here are their phenotypes. It's all, fine. All right. Here's for the ability to taste a chemical called PTC. Same thing being tracked. All right. Or same sort of thing going on here. I used to bring in PTC paper. You might have done it in a previous class that you took where it's just tastes really bitter. They'll pass it out to the class, like a little test paper. You put it on your tongue, you're like, oh, it's really bitter. Some people can taste this compound, some people can't. All right? Cool. So this individual cannot taste it, this individual can taste it. Now, this is nice to indicate like the filled in symbol and the trait that's being tracked can change based on what pedigree you're looking at. So you gotta make sure you read the pedigree chart. Like you gotta understand the setup. This is indicating like, oh, filled in symbol means not a taster. Can't taste this. All right? Cool? So, in pedigree analysis, if the genotypes aren't present, it can be a little more tricky to track, what genotypes the offspring will have, okay, or what genotypes the offspring have. So like this individual right here, we don't know if they are homozygous dominant or heterozygous. They have the dominant phenotype, and it's possible, because both of their parents had the dominant phenotype, that this individual received a dominant allele from both parents, or they received a recessive allele from one and a dominant from the other. All right? It's more likely that they're heterozygous than homozygous dominant. It's twice as likely, because there's two ways that could happen, right? There's only one way you could be homozygous dominant, but we don't know, okay? So we're going to talk about pedigree analysis in more complex patterns of inheritance, specifically with X-linked characteristics that are on genes that are only found on the X chromosome, and with blood types, which are an example of codominance or multiple alleles, where there are multiple versions of a gene that all get expressed if present. We're going to do that and kind of skip over the basic pedigree analysis, except that I'm showing you here, like here's what basic pedigrees look like. All right? So last slide, and then we'll end for the day, get into complex patterns of inheritance tomorrow. Autosomal disorders, okay? Autosomal disorders are on the chromosomes that are not the X and the Y chromosome. The X and the Y chromosome are called the sex chromosomes, okay? The rest of the chromosomes are called autosomes, all right? So autosomal disorders are those that are encoded on the non-sex chromosomes. And they're all going to follow rules of inheritance, based on what we've already talked about. Sex-linked or X-linked chromosomes follow different rules because some people have two X's and some people only have one X chromosome. So, follows different rules. Some autosomal disorders, though, recessive versus dominant disorders. What this means is a recessive disorder, okay? You need two copies of the recessive allele in order to express the disorder. For example, albinism, all right? So if you have albinism, there's a lack of pigment in your skin, hair, and eyes. It's due to an enzyme not being formed that's involved in pigment deposition, all right? So you've got all the genes present for having a certain hair color, a certain skin color, a certain eye color, but you don't have the enzyme responsible for putting pigment into things. If you're homozygous recessive, you can express the phenotype of having albinism. All right? Fair enough? This means that there can be carriers for albinism, heterozygotes, okay, that don't have albinism, but if they have an offspring, that offspring has a one out of every four offspring they generate, if they're both heterozygous, all right, would have albinism. All right? It's a recessive disorder. It is an autosomal recessive disorder. These are examples of dominant disorders, where you only need one copy, all right, of the allele in order to express the disorder. So achondroplasia, which can lead to one form of dwarfism, all right, is a dominant autosomal disorder. That means, right, that if you have one copy of the allele, then you express the phenotype. But two people with dwarfism, right, or achondroplasia specifically, could have an offspring that does not have that characteristic, if they're both heterozygous. 1 out of every 4 offspring would be, of normal height or would not have achondroplasia. Anyways, there's a lot of cool examples of this stuff. I pushed you about 4 minutes into your dang lunch though, and that's kind of a no-no. But I really wanted to get here to say that we're going to start our complex patterns of inheritance talk. So, not pure dominance, not pure recessive characters, not purely autosomal. We are on slide 100. That means that we must get through 50 slides tomorrow. We can do it. We've got almost 2 hours, right? So I just need to be churning through slides. We'll have a fun time. I'm ending the recording. Here we go.